Dear students of MSc 4th SEM, as you know, due to Corona crisis and lockdown, now we are teaching with the help of Google Classroom and video and audio, etc. I hope you all are stay at your home and are safe and following the guidelines of WHO as well as Government of India in such a crisis of Corona in India. Dear students, today we will discuss about the DNA sequencing methods. As you know, DNA sequencing is the sequencing of nucleotides. DNA consists of 4 types of nucleotides, that is adenine, guanine, thymine and cytosine. And the order of these nucleotides determine that what are the sequence of their genes or codons. So by this, the DNA sequencing method, we determine the order of nucleotides in a DNA segment. Generally, 2 methods are very popular with regard to the DNA sequencing. First is Maxam and Gilbert method, and second is Sanger dideoxy methods. Today we will discuss about both the methods in detail. First of all, we will start with Maxam Gilbert method. The chemical cleavage method divided by the Maxam and Gilbert starts with a DNA that is labeled at one end of one strand with phosphorus 32. First of all, DNA is labeled with P32, then polynucleotide kinase is usually used to add P32 at the end of the DNA. The labeled DNA is then broken preferentially at one of the 4 nucleotides. The conditions are chosen so that an average of 1 break is made per chain. Each broken chain yields a radioactive fragment extending from the label P32 to one of the positions of that base, and fragments are produced for every position of the base. For example, if the sequence of a DNA segment is guanine, thymine, cytosine, cytosine, adenine, guanine, thymine, adenine, 5' to 3'. Then the radioactive fragment produced by specific cleavage on the 5' side of each of the 4 bases would be cleavage. If we cleavage at adenine, that is here. If we cleavage at adenine, then P32 is labeled at the start of the DNA segment, and after cleavage, a segment of P32, guanine, thymine, cytosine and cytosine will be produced, whereas another segment GTA 3' will be produced. If cleavage takes place, या breakdown of the DNA takes place at guanine, then P32 GTCCA, that is here G, CCA will be produced, and if cleavage at cytosine takes place, only GT will be produced as a segment. If cleavage takes place at T, that is thymine, only G, guanine, with phosphorus 332 will be produced. The fragments in each mixture are then separated by polyacrylamide gel electrophoresis or PEG, PEG, which has the capacity to resolve DNA molecules differing in length by just one nucleotide. Next is to look at an autoiodogram of the gel. For the above result, the DNA is specifically cleaved by the following reagents. First, purine are, that is adenine and guanine, are cleaved by dimethyl sulfate. Which methylates adenine at N3 and guanine at N7. The glycosidic bond of methylated purines are broken by heating at neutral pH. The sugar backbone is broken by heating in alkali. Now pyrimidines, that is cytosine and thymine, are cleaved by hydrazine. Their backbone is cleaved by piperidine. The second method is Sanger dideoxy method. In Sanger dideoxy method, sequencing DNA polymerase first is used to copy a particular sequence of a single strand DNA. The synthesis is primed by a complementary fragment, and in addition to the 4-deoxyribonucleoside triphosphates, the incubation mixture contains a 2,3-dideoxy analog of one of them. The incorporation of this analog blocks further growth of the new chain because it lacks the 3-hydroxyl terminus needed to form the next phosphodiester bond. Hence, fragments of various lengths are produced in which the dideoxy analog is at 3 end. 4 such sets of chain terminated fragments are electrophoresed and thereby autoradiographed. These are 3 phosphates. It is the structure of 2,3-dideoxy analog. The 3' stage, DNA to be sequenced, 5' structure, DNA polymerase DTP, DTTP, DCTP, DGTP, dideoxy analog of ATPs. In such a way, we can sequence the nucleotides of DNA, and we know that what is the sequence of nucleotides in a particular fragment of DNA. Thank you. Okay.